Hello students, today in this session we are going to discuss about the regulation of gene expression. As we have discussed the formation of DNA to RNA and RNA to protein. Now what is mean by gene expression? It means the information that the DNA had stored in a living cell, in a living system, that was transmitted in the form of mRNA. Now, mRNA has to form protein. It means that the gene has been expressed. As we have discussed, the process of translation where the protein synthesis take place inside the nucleus. Now, once the protein has been synthesized, it has to leave the nucleus and it has to go to the cytoplasm. So in this session we are going to see how can the gene expression can be controlled, or how is it regulated in the living system. Now if you see in eukaryotes, there are 4 ways in which the gene expression can be regulated. The first one is transcriptional point. Transcriptional level. It means that when DNA give rise to mRNA, it means that there is transcription. Okay, so this is if you stop the formation. Okay, when there is a formation of your primary transcript, formation of primary transcript, which we also call mRNA, or you can say as hnRNA, heteronuclear RNA. So you can check, okay, the gene regulation or gene expression in this point. The next is your RNA processing. So what is RNA processing? We have already discussed that. Like splicing, okay, tailing, capping. So there are methods of modification, right? So in RNA processing, we can also check. So that is your splicing. Okay. Now third can be, you know that once the protein is synthesized, it has to transfer to the cytoplasm. So during that time, the gene, okay, the regulation of gene expression can also be checked. That is transfer of mRNA to, that is from nucleus to cytoplasm. Point number 4, that is once the protein is synthesized, okay, during the process of protein synthesis, we can regulate it. Okay, so that is translational level. So in eukaryotes, this is for the case of eukaryotes, like mammals, human being. Okay, so these are the 4 ways in which the gene expression can be regulated. Now, if you see prokaryotes, like example like E. coli, which is a bacteria. So in eukaryotes, if you see, there is a polycystonic structures or polycystonic genes, right? So in eukaryotes, they have a specialized way of regulating. Okay, so that specialized mechanism is done with the help of accessory proteins. It means that regulation of gene expression is performed by the help of accessory protein. Regulation by accessory proteins. In the case of prokaryotes. Now, just say that E. coli, you know, E. coli is a bacteria, right? So it's a prokaryote. Now, they produce a special enzyme called as beta-galactosidase. Beta-galactosidase enzyme. Okay, I'll just write E, beta-galactosidase enzyme. Now, when this enzyme is produced in a living system, in a nutrient media, if there is a lactose, these bacteria, they will produce enzyme called beta-galactosidase. In a media means the nutrient medium where bacteria needed to grow. Okay, so in this media, if there is a lactose, now what is lactose? Lactose is a disaccharide, right? Disaccharide. Now this lactose is broken down into monosaccharides, that is glucose plus galactose. So in a nutrient media, if there is a lactose, then E. coli, okay, they undergo gene expression and then they started producing beta-galactosidase enzyme to degrade, okay, to break lactose to glucose and galactose. So this is a mechanism, and if there is no lactose, then it does not, means beta-galactosidase enzyme does not produced. It means that in a simple term we can understand that the gene regulation, okay, gene expression is regulated because of the environmental condition. Okay, first point can be environmental conditions. Environmental conditions. Second can be metabolic activity, metabolic. And the third one is physiological. Physiological. So these are the three simple factors by which, okay, the expression or regulation of gene expressions are done. And this can be done with the help of specialized protein called accessory protein, or we also call these as regulatory protein. Regulatory protein. Now this regulatory protein can be of two types. One is positively. I'll just write RP, regulatory protein. So it can be positive. So if it acts positively, they are called activator. And if they act negatively, they are called as suppressor or repressor. Okay. Now, in prokaryotes, there are three specialized genes. Prokaryotes, there are three specialized genes. The first is said to be your promoter gene. Second is operator gene. And the third is called structural gene. So the combination of these three, if there is a promoter gene, operator gene and structural gene, it is considered as operon. It is named as operon. Okay, operon is promoter gene. There will be one promoter gene, there will be one operator gene, and there will be one structural gene. Now, operon is of two types again. Just go like that. Okay, so it is said to be inducible. Now, the meaning of inducible is normally off. Normally off. So this system will remain off, but when it is required, they will automatically on. Okay, when it is required, they will on. So this is a system where if there is a nutrient media, for example, if there is a nutrient media, the operand will be, they will on the system, but normally they remain off. So that is said to be inducible. Suppressible means normally they remain on, normally, but when it is required they will off the system. So example, in case of inducible, you can take an example of Lac operand, and this operand is there in your syllabus. And example for your suppressible operand is tryptophan operand. So now we are going to discuss the detailed system, okay, the Lac Operon system for the regulation of gene expression found in prokaryotes. Lac operand. I'll just make a nucleus, use a nucleus, just think that, and there is a Lac operand, okay? It has a promoter, P, I'm writing as P. It has I is said to be inhibitory gene, okay? And there will be one more promoter, then there will be operator gene, and then there will be structural gene, Z, Y, and A. Now, this is said to be promoter. I'm writing as P is equal to promoter. I as inhibitory gene, okay, promoter gene. Then O as operator gene. And we have ZY. Z is said to be structural gene, okay, but it will produce beta galactosidase enzyme. Y will produce permease, and A will produce trans-acetylase. So these three are considered as structural gene. So this is the structure of Lac operand. Now, just think that the system is on now. Now, when the system will on? Only when in the surrounding, if there is a substrate, okay? If the E. coli was provided with a nutrient media, lactose, normally the system was off. Lac operon system was off. Now, when the system was off, let's see what happened, how the system works, okay? Now, off means there is no substrate, means absence of lactose. So you can consider lactose as inducer. Okay. Now if there is no lactose, then what will happen? You know that for the synthesis of your mRNA, which enzyme is responsible? RNA polymerase. So this RNA polymerase will bind in the promoter region. Now as it starts moving, there is a gene called as inhibitory gene. Now this gene will produce, as it is a process of transcription, then it will produce mRNA. But this mRNA is known as repressor mRNA. And this repressor RNA will produce repressor protein. Just said that this is our repressor protein. Okay. So as the RNA polymerase started moving from promoter to inhibitory gene, it undergoes transcription, and it produced repressor protein. Now this repressor protein will go and bind to the operator region. Okay, will bind at the operator region. Just say that. Okay, this is the red mark which I made it. So this will be the place where there is the operator region, the repressor protein will bind. Now as the RNA polymerase continue to move, since there is a repressor protein, so what will happen? It cannot synthesize these enzymes. Since our process of translation, it's an energy dependent process, so if there is no lactose in the surrounding, why they need to produce these enzymes? Okay, so the system remains off by producing repressor protein, and this repressor protein you can say as accessory protein or regulatory protein. Now in the second condition, okay, that this happen, the Lac Operon is remaining off when there is no lactose in the surrounding. In case 2, now there is a lactose. Okay, there is a lactose in the surrounding, in the nutrient media. Now what will happen? This Y gene, which it produced permease, some amount of permease will already remain in the cell inside the nucleus. Okay, so this permease will provide diffusion. With the help of diffusion, just say this is our lactose. Okay, with the help of diffusion, they allow the permease. Okay, the permease will allow the lactose to enter inside the cytoplasm. After entering, you can also say as permeability. Permeability. It provides permeability. Bility. Okay, permeability. Now, once it is entered inside the cytoplasm, certain modification has been done, and they produce allolactose. Allolactose is just the modification of your lactose only. Once it produces allolactose, now what happened? Very interesting thing will happen now. Just say this repressor protein will leave this operator region and bind to the allolactose. So this is your repressor protein. See, the repressor protein leaves the operator region and then they bind to the allolactose. It means that the repressor protein have higher affinity towards the allolactose. Okay, so in a condition, if you see that if you went to any of your friends' party, okay, as a birthday party, you can find that when you meet your best friends, okay, maybe you may came before, but when you see your best friend who you considered as the most important one, you leave other one and then you approach to your best friend. So it means that that person has higher affinity towards you. Okay. So as the same thing, like this example you can take here, that repressor protein leave the operator region and then completely bind, okay, with the allolactose. Now if the repressor protein leave the operator region, what will happen? The RNA polymerase will move on. As it moves on, you can see that the structural gene will transcribe and they will produce those useful enzymes. In that way, once these enzymes are produced, what will happen? Beta-galactosidase, okay, will degrade this allolactose to glucose and galactose. In that way, okay, the system, the Lac Operon system works in prokaryotes. And once the allolactose is completely degraded, what will happen again? The repressor protein will go and bind to the operator region again. Now I'm going to repeat the same process, okay? Means once the allolactose is modified and produced, what will happen? The repressor protein will come and bind because it has a higher affinity toward allolactose. Okay, it means if the repressor protein leaves this operator region, what will happen? The RNA polymerase will go for gene expression. It means that the Z gene will produce beta galactosidase, Y will produce permease, and A will produce trans acetylase. So these are three enzymes. It means that once the repressor protein binds to the allolactose, and this condition will happen when in the surrounding if there is a lactose. See, the repressor protein binds toward the allolactose. Then RNA polymerase will start moving. We have already discussed the process of transcription. Okay, it means that RNA polymerase will move, and then with the help of translation we get these three enzymes. So once these enzymes are produced, this allolactose will be broken down by the beta galactosidase enzyme. It will degrade and it will give glucose plus galactose. Right? Now, since this allolactose has been degraded, has been broken down to these monosaccharides, now the repressor protein will go and bind to the operator region. It means that the Lac Operon is off automatically. So this is a system, Lac Operon system, that is found in prokaryotes, was discussed by Jacob and Manot. This system helps us to understand how the gene expression has been regulated in prokaryotes. Now in the next topic, we are going to discuss about Human Genome Project. Now, the name itself is a very big project, right? So we call this as mega project. Why mega project? Okay, it was started in 1990, okay, by US, okay, it is US Department of Energy and then the National Institute of Health in 1990. Now, they gave this mega project name because as we know that the human genome has how many base pair? 3 into 10 to the power 9 base pair. Now, each base pair has estimated, okay, is an estimated US dollar of 3 each base pair, okay. Per base pair it is $3. It means that for 3 into 10 to the power 9 base pair it is coming around $9 billion. $9 billion, okay. That is the estimated cost. Now, if they collect the data, okay, of the DNA, then they cannot be stored in a books. For example, in one page, 1000 letters has to be written. The question was, why we cannot store this data in the form of written form, okay? Why they cannot be? So, since we need, in order to store this data, we need 3,300 books which have 1,000 pages. So it will be very difficult to do that. So they need computer analysis, okay, and with the help of computer data, a new branch of biology was started, and that is called as bioinformatics. Bioinformatics. So when we say bioinformatics or Human Genome Project, it is related with DNA sequencing. For example, two human beings, okay, we have all the similar structure, all the similar organs, maybe the size may be different, right, the height may be different, but the structure that is needed to be considered as a human being, every human being have. But what makes them differences? Okay, it means that there is a differences in the DNA sequencing. So they started differentiating the DNA sequences of human beings, of different human beings. Next thing was they want to complete this project within 13 years. Okay, and the first, that is 2003. Okay, in 2003, Human Genome Project, that was the estimated or the target to be completed. Now, they had approached two methods. The first approach was means methodology. The first one was they took the coding region. It means that sequence of a coding region. As you know that DNA have coding and non-coding region. And if you don't know, please go and check the structure of mRNA, which I have discussed in the previous class. So in the first approach, they have taken the coding region, and in the second approach, that is called as blind approach. Okay, and that is they took the entire DNA, that is coding plus non-coding. That was entire DNA. Okay. Now this sequencing, the sequencing of the coding region, they said as EST. What is EST? Express Sequence Tag. Okay. And this was known as the sequence annotation. Okay. Sequence annotation. So when someone say what is sequence annotation, just say that it is a sequencing of coding and non-coding region. And if it is express sequence tag, then say that it is a sequencing of coding region. So these two approach they have applied. Now for the sequencing, what they did was, for DNA sequencing, okay, in a brief we will see, that is, first of all we need DNA, so they isolate the DNA isolation. They have isolated the DNA. Then after that they have cut. They have cut it with the help of enzyme called as endonuclease. And they got fragments, okay. So many fragments were found. So this endonuclease we also call as restriction enzyme. Okay, there starts the genetic engineering. Okay, and once these fragments was developed, means if this is your DNA, then what will happen? This restriction enzyme will cut into many small fragments. Okay, like this. Now once the fragment has been formed, they multiply into clones. They produce clones. How they produce clones? With the help of vectors. And they use two vectors. Okay. Now you may be confused. What is vector? Okay. Actually, vectors are the I will say machines. Okay. They helps to multiply. Okay. If I have 10 fragments, then I want 100 fragments more. Okay, so you cannot synthesize DNA again and again. It will take a long process. So if I have a fragment, why don't we produce the same fragments, okay, artificially in a lab? And those we said as cloned, and this can be done with the help of a vector. Now, two vectors were used. One was bacteria, and that artificial vector was BAC. BAC means bacterial artificial chromosome. And they used yeast. You know what is yeast? We use in a baking process, right? So yeast, example was YAC, yeast artificial chromosome. They used these two vectors and they produced clones. Once the clones were produced, they started DNA sequencing, okay? Now, they started DNA sequencing of those fragments. Now, it was developed by Frederick Sanger. Frederick Sanger, S-A-N-G-A-R. So, Frederick Sanger had developed a automated DNA sequencer. Okay, automated DNA sequencer was developed by Frederick Sanger. So based on this, they sequence the DNA. Okay. Now, he was also credited, okay, for the sequencing of amino acid in protein. Okay, let me consider one example with this, because that point is very important. If you know that if I have a protein, it means that there are amino acids. Now, just say that there is insulin, and for the insulin enzyme, just assume, okay, there is a phenylalanine, one amino acid, okay, then another leucine, for example, another amino acid, then just say that cysteine, another amino acids, okay. Now, how can we know that insulin have these specific amino acids? So, if I know that I need 20 amino acids, for example, to make insulin, and if I know the sequence of that amino acid, then insulin can be produced artificially in the lab. So this is an advantage, you know, like the application of the Human Genome Project. If you know the sequences of DNA, for example, my sequence is ATGC, 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 okay? Your sister sequence, for example, my sister sequence is, for example, ATGC, ATGC, and C instead of G, okay? Then, if my sister gets a sickness, then how can we know that? We can able to know because of that one sequence that make difference and that may cause genetic diseases to other human beings. Okay, so knowing about the sequence the credit goes to Human Genome Project, which was done by US Department of Energy, okay, in 1990. After that, they have gave silent features, some of the important silent features. I'm not going to discuss everything, but some of the important points I have already written at the board. You can just see that. After the project was over, they said that there were 3,164 million nucleotide base pair in a human genome. Average gene for every human being is around 300 bases. And the largest human gene, that was dystrophin, which have 2.4 million bases, okay? And it is also says that less than 2% code for protein, okay? Less than 2%. It means that if you have a DNA and you have a coding region, and that coding region, only 2% will code for production of protein, okay? There is said that around 100 to 1000 there is a repetitive sequence, and it does not involve in the formation of protein. For example, repetitive C, and it does not help for the production of protein. It is also said that the chromosome 1, okay, the highest content of gene that will found in chromosome 1. As we know that we have 46 number of chromosomes and 22 pairs, right? So 22 is called what? Autosome. X and Y chromosome we have. For female, we have XX chromosome. For male, we have XY chromosome. And Y chromosome contain the least number of genes, that is 231. Okay. And another point they have said that there is a SNP. SNP means single nucleotide polymorphism. And this single nucleotide polymorphism has been found 1.4 million at different locations. Okay. So these are some of the important features of Human Genome Project. Now, to complete the chapter, that is molecular basis of inheritance, we have only one topic, that is DNA fingerprinting, which we will discuss in a next class.